My name is Alan Taylor, and I'm Professor of Seed Science and Technology at Cornell Agritech. My laboratory has been involved in hemp seed science and technology research since the Cornell Hemp team formed in 2017. What I'm going to speak about today is on our work on the research and development of hemp seed treatments. Gary Bergstrom is my cooperator in plant pathology, as his laboratory is supportive in helping characterize the pathogens associated with disease seedlings from our research plots. We also have field trials at North Dakota State with Burton Johnson and Chuck Johnson and John Fike at Virginia Tech. So the focus of our work is on application technology to begin with, to have good methodology for seed treatment and coating technologies. This is a laboratory scale unit called a rotary pan technology, an R6 for a 6-inch diameter. This same technology is used for commercial scale equipment. So anything that we do in the laboratory can quickly be scaled up to a larger. An issue that we had early on is that we need to ensure that we have uniformity of application over the seed surface and from seed to seed. We need to protect each seed from attack of the soil-borne pathogens at time of sowing. We'll take a step back and look at the biological market, as there's a number of materials. Now, this is not restricted to seed treatments. These could be foliar applications or soil applications and furrow applications. We have biofertilizers and biostimulants, which we're not going to be talking about, even though my laboratory is working with biostimulant seed treatments. But we're going to focus on biological control products, in particular the biopesticides. So we're going to kind of focus in on that area. So under the biopesticides, we have biochemicals. They may be kind of a mineral based. They can be either formed from copper or phosphates. We have examples of those. Microbials, we normally think of a microbial as a biopesticide, but they're not the only materials. But in this case, we are using specific seed treatments from Bacillus and Trichoderma and with others, and these are commercial formulations that we are testing as seed treatments. So this is our list of 12 seed treatments that we have. Those in green are the biologicals. The first thing we have to do is test the seed treatment application rate. We want to ensure that there's no phytotoxicity from the seed treatments. Those in red are the biochemicals. Safeguard is an organic copper. The Prudent is a phosphate material. And then the group at the bottom in blue, Apron XL Maxim, you probably recognize, as these are widely labeled on vegetables and field crops as seed treatments. So first, let's look at what happens to seeds after they're planted. We start off here in the laboratory to look at hemp seeds germinate very rapidly. So in this pathogen-infested soil that we have the soil sprinkled over on top during the germination process, after 4 days, the seeds germinate very rapidly. After 7 days, many of the seedlings have died or are dying, and by 10 days, non-treated seeds are killed. So this is a way that we can use a laboratory screening method, taking advantage of a naturally pathogen-infested soil to go ahead and to test these seed treatments before we actually go to the field. But our primary interest in showing you today is the field data. This is we have two field studies at Cornell. The first one planted on June 12th. Again, now we can see how this color coding is going to help us. The black bar on the left is the control, non-treated. You can see all the green treatments, none of the biologicals doing any better than the control. However, the Safeguard, the organic copper, the Prudent, the phosphate doing quite well and as good as one of our best Apron Maxim treatments. So this looks very promising. We have a second study also in Ithaca this year, planted on June 26th. Again, the same trend is the biologicals not performing any better than the non-treated control. We are seeing improvements with both the Safeguard, in particular the Safeguard and the Prudent. And again, it's comparable to our best Apron Maxim treatment as the chemical seed treatment. This is where Gary's laboratory was instrumental in supporting this project. We would see the symptoms in the field as far as diseased seedlings. His laboratory has done the characterization of what pathogens are associated with those diseased seedlings. So we can see here in the picture on the photo at the bottom on the left, indicative of those testing positive for Fusarium species. The picture on the lower right having Pythium species present. Gary's laboratory is still ongoing work to characterize the species of these, along with other samples that we have provided to their lab. As I said, this is a multi-state project, and being a multi-state project is good in that we do not want to simply have seeds that are tested in Cornell that only have good control in the Northeast. So we've expanded the testing. We have seed testing at Virginia Tech in the southern region, and then North Dakota State in the north central region. Again, the same color coding and the same scheme as we had in our previous slides. We see Safeguard and Prudent doing very well, and this is from one study we have. There'll be one more study coming with data this fall from Virginia Tech. And then North Dakota State, in this case, we're seeing the chemical seed treatments doing quite well in this one across the board. But still, we're seeing, for example, Safeguard doing a nice job. So we're gathering information now, and the very good picture is that we're seeing some consistency in our results from field to field and from location to location within the regions of the United States. So let's go ahead and summarize where we're at with this. Our biochemical seed treatments, the Safeguard, which was the organic copper, and the Prudent, which is a phosphite, had good efficacy in all of the studies, the data that we have in hand at this point in time. The chemical seed treatments, Apron XL and Maxim, still had good efficacy, and again, these are tried and true chemical seed treatments that are widely labeled. Unfortunately, after 2 years of work where Syngenta was initially very interested in this project, Syngenta is no longer providing support for hemp on any particular application. So it could be Syngenta herbicides and things like that. They're not going to be supporting those hemp labels in the United States, which is a big setback. So we really need other chemical seed treatments from another company to test, and we're looking into that at this time. Finally, the biologicals, and we've done now 2 years in a row trying to test a good range of commercially available and used biological seed treatments. Simply were not effective in our study to control damping off. So I want to thank people, first of all, in my laboratory, Hillary Mayton. Hillary kind of headed up all of the field studies that you had, as well as gathering data from the other states and then summarizing some of the data in the graphs that you're seeing. Michael Luce in my laboratory has done all the seed treatment application, a lot of the in-house testing as far as germination, soil testing, and our soil bioassay. Masi's done a lot of the photographic work and other support work for this project. So very nice team that I'm very fortunate to have people working with me on this project. In Gary's laboratory, Jen Starr was really instrumental in working with Hillary, taking those field samples and doing the characterization. And Kevin in Gary's lab is helping at the molecular characterization so we know what species are involved. Again, we work very closely with the company registrants. In other words, the companies that actually are producing these. We're working directly with their R&D people to make sure that we're testing the best biological, chemical, biochemical treatment that they would have available for this project. And finally, I want to thank support from IR4, but in particular for New York Ag and Markets for their continued support for this and for other Cornell Hemp projects. Thank you very much.